Hi, my name is Justin Chen, and this is my journal club presentation. The article I've chosen to present is Ketogenic Diets Alter the Gut Microbiome, Resulting in Decreased Intestinal Th17 Cells by Tianang and their associates. I want to open this up by saying what we eat is more than just getting enough energy for the day. Our gut microbiome is a very complicated system that is a very exciting frontier of science and medicine, and I think this article is a great example of that, as it reveals the relationship between the ketogenic diet and the immune system through alterations of the gut microbiome. It has been previously established that the ketogenic diet not only induces changes in our blood by increasing levels of ketone bodies, but also induces changes in the gut microbiome. There have been previous studies that looked at the correlation between gut microbial communities and diets like the ketogenic diet, but there were confounding variables when it came to changes in the macronutrient profiles, such as going from 50% to 15% carbohydrates or 30% to 15% carbohydrates. And there wasn't a study that discovered the specific mechanism that was at work. Moving on to our brief overview for this presentation, we will be looking at what is the ketogenic diet and how can the results of this study be extrapolated onto other diseases involving T helper cell 17. We will go over the results of this article's human, mouse, and in vitro study to find the mechanism that drives the reduction of T helper cell 17. And finally, the results of this article that dives into the differences between a high-fat diet and a ketogenic diet. So let's start by defining one of the two important terms that we'll be seeing throughout the presentation. If you're interested in nutrition and dieting, you've probably heard of this before, but if not, that's fine. The ketogenic diet is generally recognized as a high-fat diet that has little to no carbohydrates. In this study, the macronutrient proportions in the human trial was 5% carbs, 15% protein, and 80% fats. But in the mouse studies, it was a simple 90% fats and 10% protein. If you've ever looked up the ketogenic diet online, you've probably found similar, although slightly different macronutrient profiles. That being said, the ketogenic diet is not just another trendy fad diet. What makes this diet so special is that it replaces the glucose with ketone bodies as the primary energy source in your body. Although not fully understood, it's believed that the ketone bodies are the reason for the ketogenic diet's benefits in multiple immune and metabolic diseases, such as intractable epilepsy. That being said, in this presentation, we will be looking at the relationship between the ketogenic diet and the immune system specifically. Speaking of the immune system, this is our second term, Th17 or T helper cell 17. T cells are an important part of our immune system as they are able to recognize and take action when something goes wrong. There are several different types, but we're focusing on the subset 17 or T helper cell 17 as it plays a major role in inflammation. Inflammation, of course, is the major way that our microbiome affects us. As you can see here, intestinal Th17 cells can induce DNA damage through inflammatory molecules to change normal tissues into tumors, studied by Fesno et al. 2024. And T helper 17 cells have been associated with immune-mediated diseases, including psoriasis, rheumatoid arthritis, multiple sclerosis, inflammatory bowel disease, and asthma, studied by Tesmer et al. 2008. Now onto the methodology of this article. As mentioned before, this article consists of a human, mouse, and in vitro study. They had a very thorough methodology section, but unfortunately, it is a bit beyond the scope of this presentation, especially their metabolite analyses. So I just wanted to highlight the fact that they use 16S rRNA sequencing to identify the bacteria. They also used Sanger sequencing when they were isolating random colonies of bacteria to find bifidobacteria, which you'll see later. But In all other cases, they used Illumina the sequence along with Chime and Data 2 to improve the quality of the high throughput reads. Here are our first figures. This is from the human study, and it's a close reproduction of a similar article, except that they introduced a control baseline diet before they had the participants switch to the ketogenic diet. The other one I'm talking about, they also did not control calories, so that was another confounding variable that is controlled here. In figure 1A, you can see the proportions of the baseline diet BD, and as it's 15% protein, 35% fats, and 50% carbs. Right next to it, KD, which stands for ketogenic diet, it is 15% protein, 80% fat, and 5% carbohydrates. Figure 1B shows the concentration of ketone bodies in each of the diets. So you can see that there was a significant increase in the concentration of acetoacetate and beta hydroxybutyrate when the participants were on the keto diet compared to the baseline diet. So this human study consisted of 17 male volunteers in multiple metabolic wards where they were fed the baseline diet for 4 weeks and then the ketogenic diet for 4 weeks. And at the end of each 4 week diet arm, their stool samples were collected. For the last 7 days, their stool samples were collected. The DNA was extracted and sequenced with Illumina to find the relative abundance of different species based on the 16S rRNA sequence. Now, in figure 1D, you can see the changes of relative abundance of actinobacteria, bacteroidetes, firmicutes, and proteobacteria, but we will be mostly focusing on actinobacteria as it contains the genera bifidobacteria. As you can see, there was a very significant decrease in the relative abundance of actinobacteria, and I think this is even better illustrated by figure 2B all the way on the right side of the slide. This is multiple species of bifidobacteria, and each line shows the change of their relative abundance per participant. Again, nothing too new or surprising on this slide, as the procedure for this was largely based on another study. But I always like to see other studies' results get reproduced. Now we are moving on to our mice studies, and they're pretty similar to the results from the human trial. Again, on the top left, you can see the macronutrient proportions of the different diets, this time consisting of chow, a low-fat diet, a high-fat diet, and the ketogenic diet, LFD being low-fat diet and HFD being high-fat diet. So now, instead of only a baseline diet to compare the ketogenic diet to, we're also comparing a high-fat diet that contains 10% protein, just like the mouse ketogenic diet. But instead of 90% fats, it is 75% fats and 15% carbohydrates. In figure 3B, once again, we're observing a significant increase in beta hydroxybutyrate when on the keto side, when on the keto diet. And in figure 3F, we see some significant changes in the microbiota across the different diets. Looking at figure 3G, it is a graph to represent the differences between a high-fat diet and the keto diet. And if you take a look at the bar way on the right, you'll see bifidobacteria, which is much lower in the keto diet compared to a high-fat diet. Even though the only difference between these two diets is the small portion of carbs, we're still seeing a pretty drastic difference between the two. So the researchers investigated next was, if you increase the proportion of fat, will these higher fat diets perform similarly to the ketogenic diet in mice? And if they continue to investigate them, would they find a specific threshold, or does it start to look more dose dependent? Which brings us onto our next slide. As you can see from these figures, even small amounts of carbs in the gut will still induce changes in the gut microbiota. Figure 4A shows us the macronutrient proportions of these higher fat diets fed to mice, and 4B accompanying it shows the still significantly different concentrations of beta hydroxybutyrate for each one of these higher fat diets. Now, I've included a small image of figure 3b from the last slide, just on the right of 4b. And as you can see, the concentration of beta hydroxybutyrate compared to the higher fat diets and the keto diet have become much more linear. And this is a change we see in the relative abundance of actinobacteria pictured at the bottom left in figure 4G and bifidobacteria pictured in 4H. What we see is that approaching very high fat diets where there's only 5 to 1% carbs still induces changes to the gut microbiota, despite it not technically being a classic ketogenic diet. But what part of the ketogenic diet produces this effect? Is it the presence of circulating ketone bodies? Is it the significant presence of lipids in the intestinal lumen? Or is it the relative absence of carbohydrates in the diet that's driving these changes? Which brings us to our next slide. Now we're getting somewhere interesting. At the top left in green, I've pictured beta hydroxybutyrate, just for reference, because in this section, the researchers used ketone esters pictured under 5A to find out that the presence of ketone bodies directly inhibits the growth of certain bacteria. They did this by supplementing both a high-fat diet and ketogenic diet with ketone esters. And as you can see in figures B, C, and D, the diets that are supplemented with ketone esters, represented by the letters HFD-KE and KD-KE, both have higher concentrations of beta hydroxybutyrate compared to the non-supplemented high-fat diet and ketogenic diet. In figure 5I, you can see the change the ketone esters make, especially for bifidobacterium and lactobacillus. The bright orange and dark blue sections are so much lower than the pink and light blue sections, respectively. Also, in figure 5J, it's a little hard to tell, but in the top right of the graph, it gives a rho value, which just tells us that the concentration of cetyl beta hydroxybutyrate increases as the amount of bifidobacterium decreases. I don't think this graph is necessarily the best representation of the relationship, as it's quite hard to read. But from here, the article starts presenting in vitro studies, like figure 5K, that uses concentrations far beyond what we'd find inside anybody. Figure 5K is a growth curve of gut microbiota, and each color line represents a growth medium with a different concentration of beta hydroxybutyrate. The top red line is none. The green line is 12.5 millimolar. The blue line is 25 millimolar, and the purple line is 50 millimolar. Again, the green, blue, and purple lines represent concentrations way far beyond what we'd find inside a person or a mouse. But this clearly demonstrates that somehow, although we don't know the mechanism why, beta hydroxybutyrate is directly inhibiting growth of gut microbiota. This slide and the next two is where everything sort of starts coming together. I know it's been a lot of information as I've taken you through this long pathway, but so far we can definitely say that ketogenic diets alter the relative abundance of certain bacteria through its ketone bodies, more specifically speaking of beta hydroxybutyrate. And this slide is where we see that mechanism. In figure A, we see growth curves again. On the left, we have beta hydroxybutyrate, and on the right, we have pH neutralized beta hydroxybutyrate. As you can see between the two, when beta hydroxybutyrate is pH neutralized, it hardly does anything to bacterial growth and definitely doesn't come close to flatlining it, even at concentrations as high as 50 millimolar. In figure D, the researchers used a pH matched treatment instead of beta hydroxybutyrate. So they pretty much used a neutral solution at the red line with 7 next to it to represent 0 millimolars of beta hydroxybutyrate, and then used progressively more acidic solutions to represent higher concentrations of beta hydroxybutyrate. And it looks nearly identical to the growth curves when the bacteria or when the microbiota are actually treated with non-neutralized beta hydroxybutyrate. So from this, the researchers now know how mechanistically a ketogenic diet can affect gut bacteria. It does it through a pH-mediated mechanism. Now, these graphs are looking a little different from the ones before, and that's because we're now looking at T helper cell populations. The researcher used flow cytometry to measure T helper 17 cells, or as you can see on the y-axis of the bar graphs in the middle of the screen, interleukin 17A positive, CD4 positive cells. It's just counting the cells that express these two molecules, which are going to be T helper 17 cells. So they're the same thing. GF in this case represents the germ-free mice. So they don't really have gut bacteria. And BA represents mice that have been monocolonized by Bifidobacteria adolescentis, which was the bacteria that was Sanger sequenced and isolated from the human stool samples. So they only have Bifidobacteria adolescentis in their gut. And it is also a bacteria that is greatly inhibited by the ketogenic diet. Going over more acronyms, again, HFD is high-fat diet and KD is ketogenic diet. So in figure A, both types of mice are on a high-fat diet. And the little box on the map has a value next to it that reflects the percentage of T helper 17 cells, which might be a little hard to read, but if you turn your attention to the bar graphs, you'll see virtually the same information. So you see SI on the bar graph, that's just small intestine. We in figure A, we see a significant increase of T helper 17 cells in the small intestine of monocolonized mice compared to the germ-free mice. But in figure B, when the two types of mice are fed a ketogenic diet, we see no significant differences between the germ-free and monocolonized mice. So what you can take away from this is that the ketogenic diet reduced the population of T helper 17 cells, which again plays a significant role in inflammation. So, and between that, of course, is the Bifidobacteria genera. So the ketogenic diet produces ketone bodies, which inhibits the growth of bifidobacteria, which in turn seems to, in reducing that, also reduces the amount of T helper 17 cells. I know that was a lot of figures to get to this point, but I hope you've been able to see the process on how we finally got here. So onto our last and final page of figures, we take a look at every diet we've seen with the mice so far, along with the T helper 17 cell population in the small intestine seen in figures A and D, but also the T helper cell population in adipose tissue in figure G. There are a few standout results on the bar graphs, especially in figure D, the high-fat diet, 89% is oddly high, but overall, across them, you can see that the mice that were fed the ketogenic diet has a much smaller T helper 17 cell population in both the small intestine and adipose tissues. So what's the big deal at the end of this? Because we're not mice, and this study only really elaborated on processes we already had an idea of. Well, the human gut microbiome can rapidly adapt to dietary changes. And in the study that's cited on screen, it had diet arms of only 5 days. And the stool samples that were collected at the end of it revealed significant changes in the gut microbiota. So even though we're not germ-free mice, we can still experience these changes ourselves. And if you've ever heard the idiom, an ounce of prevention is worth a pound of cure, you'd realize that a diet that can reduce inflammation, and as it is a diet, it would be chronic. It'd be reducing chronic inflammation. It could be hugely beneficial, especially to those predisposed to immune disorders, metabolic disorders, and even reduce risks of cancer. And by that understanding, also by understanding the mechanisms that diseases and treatments work, we can extrapolate the results of this study to pose future research questions to see how it might work for other disease processes that involve T helper cell 17, or what other diseases might be affected by the ketogenic diet. Before I leave you with a slide of references, I'd like to present you all with some optional discussion questions. This first one actually relates back to figure 5I on a previous slide. So, how might a decreased lactobacillus population affect us while eating a ketogenic diet, as that wasn't heavily explored? How well might the results, how well might the results from the mice studies translate over onto humans? Regardless of their efficacy, how practical is it to recommend or treat somebody with a higher fat or a ketogenic diet, especially given the palatability of it? And are there certain disease states that can be exacerbated or worsened by the microbiome composition of a ketogenic diet? And finally, is there a comfortable way to increase ketone bodies in our bodies without having to enter ketogenesis via ketogenic diet? Thank you for your time, and these are my sources.